Hello, and thank you for your interest in our poster. This poster is focused on the investigational drug SM-88 that is currently in clinical trials for several different cancers. SM-88 is a chemically modified D-L-racemic formulation of tyrosine designed to leverage the cancer's unique consumption of non-essential amino acids and disrupt various cancer mechanisms. Both D and L isomers of tyrosine have been shown to be taken up by cancer cells via the transporter LAT1, or L-amino acid transferase 1, that is highly overexpressed in cancer cells. A majority of cancers are known to overexpress these transporters, and this biology has been leveraged in cancer imaging for over a decade with the use of radiolabeled amino acids for positron emission topography. Clinical data generated for SM-88 to date has been very encouraging, including confirmed responses across 15 different tumor types with a well-tolerated safety profile. Additionally, SM-88 has shown to inhibit the production of catecholamines that can have an important role in both systemic and tumor immunity. Data from two xenograft studies are presented here. Our initial study used the ACT-116 human colorectal cancer line implanted subcutaneously into nude mice. This study examined a range of SM-88 doses delivered by oral gavage. While there are some modest reductions in tumor size observed in the lower doses, the most meaningful anti-tumor effects were seen at the highest dose studied. Our second xenograft study used the PAN02 mouse pancreatic cancer cell line implanted subcutaneously into C57BBL6 mice. Because one of the goals of this experiment was to look at immune alterations, we conducted this experiment in a fully immune-competent mouse. In the PAN02 xenograft study, we observed a significant anti-tumor effects with SM-88 at a dose of 75 milligrams per kilogram delivered IP. Figures 2A and 2B display the study results, and the end-of-treatment excised tumors in Figure 2B give an idea of the proportion of tumors in the study that remain below the 500 milligram cubed, or about 2.5% of the total body weight of these mice. Twice as many tumors from the 75 milligram per kilogram dose group remain below this threshold compared to the control group. Moving to the immune analysis, from the PAN02 xenograft studies, 5 animals were randomly selected to have their tumors examined by flow cytometry using a 16-marker panel. These early results were quite interesting, with changes observed in the number and the proportion of several immune populations, as well as that are well recognized for their role in tumor immunity and response to current immuno-oncology therapies. First, there appeared to be a reduction in the M2 macrophages that resulted in an increase in the M1-M2 ratio in these samples. This data supports our hypothesis that the impairment of catecholamine production by SM-88 may reduce polarization of tumor-associated macrophages towards the M2 phenotype. Second, there appears to be a similar reduction in CD4, but not CD8 lymphocytes, and a subsequent decrease in the CD4-CD8 ratio in these samples. With regards to cellular effects, we examined the potential alterations of autophagy, starting with the expression of the LC3B protein. We paired this with the marker P62 that is normally inversely expressed during autophagy. We noticed that LC3B expression increased in several cell lines with SM-88 exposure, suggesting either a potential stimulation of autophagy or an impairment of a later phase of autophagy. Increasingly, P62 remained high or increased with the dose and may suggest a disruption of the autophagy. We have several additional studies ongoing to further examine how SM-88 may interfere with some portion of this important but complex survival mechanism. And finally, SM-88 appears to have a dose-dependent increase of reactive oxygen species production across several cell lines. Possible sources of ROS will be explored in future experiments. In conclusion, we found that single-agent SM-88 displays anti-tumor effects in both nude and immune-competent mice. SM-88 appears to have a broad impact on the immune dynamics of the tumor microenvironment, including reductions in M2 macrophages, CD4 T cells, and Tregs. And finally, SM-88 appears to disrupt autophagy or similar cellular processes that are important for cancer cell survival and increases ROS. Our ongoing future research will initially be focused on determining more details around autophagy interactions, including what steps in the process may be affected, as well as similar pathways like mitophagy, gaining a greater detail on the immune alterations with SM-88, potentially signaling pathways that might be involved, so IO combinations with SM-88 could be explored, and additional dose optimization and testing with other xenograft models. Thank you again for your interest, and please contact contact us by email or call us as indicated on the poster, and additional information can be found on our website